From the first two talks, we learned that it's very important that this beta-catenin should be transcriptionally active, and is important then to look at the transcriptomic profile, as we call in the scientific field. So to discuss that, I would like to invite Dr. Florencia Haase to give a talk on transcriptomic profiling of AAV9-CTNNB1 gene therapy, which is URBAGEN, in patient-derived organoids. The floor is yours.
Thank you. Okay, thank you very much. And thank you to the organizers for allowing me to present our research today. So, as mentioned, I will talk about the transcriptomic profiling of AAV9-CTNNB1, or also known as URBAGEN, in patient-derived organoids. So, I guess everyone knows that there is a big challenge in translation from what we do at the bench and then what we translate into the clinic. There's a lot of things that we can do at the lab that then we try in mice and it works, and then when we go into humans, the effect that we see in murine models, for instance, is not the same. They are not efficacious. So there's, I think, one of the challenges is not finding a perfect model, but is being able to build enough evidence to be able to move forward safely into patients. So I really like this quote from a statistician, George Box, who said, "Remember that all models are wrong. The practical question is how wrong do they have to be not to be useful?" So with that in mind, in the package or in the evidence chain for URBAGEN, we decided to include the use of organoids. So brain organoids are human developmental models. They are also sometimes called, I think mistakenly, mini brains. They are really more controlled human developmental models, I think. And they are basically, what we do to generate these models is we get patient cells, so we can get fibroblasts or PBMCs. Then using different transcription factors, we convert them into iPSCs, or induced pluripotent stem cells, which can virtually differentiate into any type of cell of our body. We then start an induction, also called neural induction, and then that's how we form these brain organoids. Now, the good thing about brain organoids is that we are able to preserve the patient's genetic background, in this case, of course, carrying the haploinsufficient mutation of CTNNB1. We are able to model early cortical development, and we've got multiple cell types, including neural progenitor cells, neurons, and glia, and if we drive them long enough, we can also see oligodendrocytes present in those cultures. And of course, they enable us to do readouts such as RNA sequencing or protein expression. We can also subject them to MEA assays, which are microelectroarray assays, where we look for electrophysiology, which would be the equivalent of an EEG in a human. So this is basically the experimental workflow that we did. So we had the brain organoids from several CTNNB1 patients. We, of course, differentiated them into organoids, and we had 3 groups. The first group is our treatment group at the top. Then we had an AAV GFP, which is just the capsid carrying a GFP fluorescent marker, and an untreated control. And then we also had 2 controls from healthy donors. We left them in culture for a number of days, and then we collected these organoids into different time points. Then from a bioinformatics perspective, what we do is get all the data from RNA bulk sequencing in this case, and then we normalize that data. And then we looked at comparisons between AAV-CTNNB1, which is a treatment, against the untreated patients and the control samples. The reason why we include the AAV-GFP is to look for changes that could be derived from the capsid itself rather than the treatment, transcriptomic changes, of course. And then we do the interpretation. In this case, the results that I will show are all at a pathway level. So we know, and as mentioned in the last two talks, that beta-catenin is involved in many things, not just in the Wnt signaling pathway. And basically, what we looked at was the Wnt signaling pathway at cell adhesion. We also look at maturation and synaptic organization, and of course, at the safety concern, which is oncogenesis in the case of CTNNB1. So I've kind of like draw this map because I will now show a lot of graphs, and I'm trying to be as interactive as I can and not boring. But basically, what we did was first look at CTNNB1 target engagement, so at the basically levels of CTNNB1 expression. Then we looked at developmental maturity, neuronal and synaptic response, the glutamatergic response, the Wnt signaling pathway, of course, cell adhesion, and then lastly, safety. So this is the first result that we got. So we have, in this case, 3 patients. They are all loss of function patients. We had 1 patient with a frame shift mutation, 1 with a whole deletion, and then a nonsense mutation as well. So in this graph, what you can see is we have the untreated samples here, and then we have the how they progress in terms of expression of CTNNB1 post-treatment. And in the gray shaded area represents the 2 healthy controls, the expression of the 2 healthy controls, and the dotted line is the mean expression of the healthy controls. So you can clearly see that after treatment, expression goes up. Then we looked at developmental timing. And as I've mentioned, we have 2 time points in this experiment. So we were looking into, sorry, whether the maturity score was related also to the induction of the treatment. So basically, in this graph, we can see untreated and treated. And if you look at time point 1 versus time point 2 in the untreated, the maturity score arrives up to like 0.25 for 1 of the patients. Whereas when we look at the treatment, the maturity score goes a bit higher, or yeah, quite a bit higher. And that's a sign that the treatment is helping in maturation. We then, for the next of the results, I will show all results from time point 2 because those organoids were more mature, so that means that they're more relevant for the disease that we are talking about. So all of these results are time point 2 based. I've also done this graph on the left, sort of like to try and make it more simple to understand the transcriptomic changes. So basically what we see here is I've grouped all the patients, so patients 1, 4, and 5 all together, and we've looked at changes post-treatment in terms of neuronal activity and synaptic function. I'm only showing a couple of those genes that represent this pathway, and as you can see, they're all moving towards the right, which means that those genes are being upregulated. In this context, so in the context of synaptic function and neuronal activity, we are looking for genes that upregulate and that are moving towards a healthier, like more of a healthy-looking transcriptomic profile. On the left, what you can see is the decoupled patients as well, and the expression of most of those genes is moving towards the positive side of the graph. This is looking at glutamatergic synapses, and again, the same concept. So here, in this graph, you can see that most of the expression of those genes for those 3 patients is moving towards the positive side, so they are being overexpressed. And again, same concept here, trying to look for, looking at all the patients together and where the treatment is driving the expression of those particular genes. Now I'll move on to the Wnt signaling pathway. So here we look at the Wnt signaling pathway and some of the genes involved there. And again, you can see that there is upregulation of those genes post-treatment. And then moving on to the results that we got for the cell adhesion. Again, we see a positive upregulation. We try to look at these results from a pathway point of view. Of course, there's genes that will not move or upregulate as much, but overall, we were looking for a pathway response, and it was significant. And now, this this is looking at safety and proliferation. So we know that CTNNB1 is involved in oncogenesis. So we were trying to look for broad oncogenic activation. So here we are looking at the opposite thing. We are looking for genes that will not upregulate. We are trying to keep them as quiet as possible in a way. And we see that patients 1 and 5 respond quite well. There's some upregulation for patient 4, but overall, there was no broad oncogenic activation. Now, I think what we can conclude from organoid RNA sequencing data is that, so we do have limitations. There's organoid batch effects, and that's why we require a conservative data set selection. That's why there's a huge normalization step. I forgot to mention, but in this case, we used patient 1, which was the patient that we had most of the data on, and we used that patient to then be able to make sense of our data and see which samples could be included or not. We also did multiple batches of differentiation or biological replicates. And of course, utilizing bulk-seq RNA as a readout, which is what I've shown today, we cannot really resolve which cell populations are driving this signal, because when we extract RNA from the whole organoid. This data set is supportive, I think, from a mechanistic point of view. We cannot, it's not, of course, a standalone efficacy package, so going back to the point of translation, everything, I think, has to be looked in context. And this data has helped us to look at target engagement, mechanistic directionality, molecular safety screening, and it helped us then to move on to what was going to go into animal studies as well as other clinical studies in terms of pathway analysis. So in overall, organoids supported construct selection because we also use organoids to select the constructs that we were then going to test in animals and in vitro. RNA sequencing showed pathway-level target engagement in the Wnt signaling pathway, adhesion, maturation, and synaptic biology. And from the safety-oriented transcriptomics, we did not see broad oncogenic and proliferative activation, as I have shown. So yeah, with that, I'd like to thank everyone. I forgot in my acknowledgement slide, but I would like to thank my lab, the Translational Vectorology Group in Australia, in the Children's Medical Research Institute, and of course, the CTNNB1 Foundation. Yeah, that's all. Thank you.